Welcome. Today we are going to learn about the synchronous culture. First, what do you mean by the synchronous culture? Synchronous cultures are the cultures in which all the cells, all the bacterial cells, they are in the same phase of their life cycle. That culture is called as a synchronous culture. Okay. Here we have learned in the normal growth curve that the bacterial cells, it is going through the four phases of its life cycle: lag phase, log phase, then the stationary phase, and then the death phase. Okay, so when you take the culture, okay, when you take the bacterial culture, at that time, in that bacterial culture, the all the cells, they may or they may not be in the same phase of their life cycles. Means some of the cells they are going to divide, some cells they are dividing, some they are about to divide, that is the cells they are the mature one. That is the phase of the life cycle of the bacterial cells, it is going to be a different. Now when a person or when we have to, okay, when we have to study the bacterial culture, what is happening into that bacterial culture, what is happening into a bacterial cell at the development of bacterial cell, or at the stage when it is dividing, when it is maturing, what is the macromolar constituents of that bacterial cells, when we have to analyze at a particular phase of bacterial cell, at that time you cannot analyze in the heterogeneous population of the cell mass of the bacterial cells. What do you mean by this? Now here it is what? It is your bacterial culture, and some cells they are mature, some cells they are divided, or some cells they are dividing. Okay, some cells they are the daughter cells, they are just form cells. So here when you look at this, all the cells, the cellular constituent of each and every cell, it is going to be different. Okay, so such a heterogeneous population, by using this such a heterogeneous population, one cannot analyze, okay, one cannot analyze the what is the cellular constituent, or what is the macromolar constituent, or how the cell, what is happening into the cell at that particular developmental stage. It cannot be analyzed into this heterogeneous population of the cells. Okay, so what has to be there? There has to be the presence of, okay, there has to be the presence of the cells, okay, there has to be the presence of the cells, okay, which are in the same phase of the life cycle. That is, all cells they should be the daughter cells, or all cells they should be the mature cells, okay, in a such a way that there should be the presence of the population of the cells. That is, all cells they has to be present in the lag phase, or they has to be present into the log. And if you want to study the stationary phase, then all the cells they should be in which phase? They should be in a stationary phase. Then you can study what is happening into that particular cell at that particular phase of the bacterial cell. So you have to obtain, okay, you have to obtain that culture which is having the same phase of the life cycle of the bacterial cell. And such cultures which has been obtained, such cultures, they will be called what? They will be called as the synchronous culture. Or you will say that the synchrony, it is obtained into the cultures. Means what? What do you mean by that synchrony? Obtained means what? The all cells, they are, all the bacterial cells, they are in the same phase of the life cycle. So why you want all the cells, they should be in a same phase? Here, when you want to study the bacterial cell, it is very small. You cannot study the single cell, okay? What you cannot study? You cannot study the single cell, okay? Because the cellular constituent of that single cell, it is a very small. As the bacterial cell, it is a smaller one. Ultimately, its cellular constituents, it is also going to be a smaller one. So here you have to study the mass of the cells. What you have to study? You have to study the mass of the cells, and by studying that mass of the cells, the study of that mass of the cells, it is going to be equivalent to, study a individual cell. Okay, are you getting? So here when you want to study the bacterial cells, here you cannot study the bacterial cells singly. Okay, so you are requiring the mass of the cells, that is the cellular mass of the bacterial cell, it is required. But as you have to study the single phase of the life cycle, you have to obtain the culture in the same phase of the life cycle, that is you have to obtain the culture into the synchronous culture. You want the synchrony into the culture. All cells they should be in the same life cycle. And also whenever you do the inoculation, in the fermentation industry, when you do the inoculation, in some of the industries there is a requirement that the cells they has to be present into the same phase of their life cycle. That's why here there is requirement of the synchronous culture. So how to obtain this synchronous culture? To obtain the synchronous culture, here there are the techniques. There are the selection techniques, and that selection technique, it is the first selection by size and age, and the second one, it is what? Selection by induction. Okay, there are the two techniques, there are the two methods by which you can obtain the synchronous culture. First one, it is the size and age. In that there are the two methods. One, it is a Hemsteller-Cummings method, that is a technique, and the second one, it is a density gradient centrifugation. Okay, so going towards the first one, it is a size and age. Now here this, it is your mature cell. Okay, this mature cell, now it is going to divide, and it is going to give you what? It is going to give you the two daughter cell. Okay, so when you look at the size of both of the cells, it is how it is a different one. The mature cell, it is of the larger size, and the daughter cell, it is of the smaller size. Okay, and the second, the age of this mature cell, it is a larger than the age of whom? The daughter cells, because these daughter cells, they are the newborn now. Okay, so here in this technique, selection, okay, selection by what? Selection by the size and the age. So here mature cell, it has a larger size, and the daughter cell, it has a smaller size. Okay, so here you are selecting what? You are selecting the mature cell. Okay, you are selecting here the mature cell. Okay, in which technique? Hemsteller and the Cummings technique. Here you are going to select the mature cells. You are going to allow the smaller cells to get drained out. Okay, now in the Hemsteller and Cummings method, here actually the filtration, it is going to be used. Okay, what is used? The filtration technique it is used. Here there is what this, it is what this, it is a membrane filter, and the membrane filter, it has been used in a such a way that the pore size, it is selected in a such a way that only the smaller size cells, they are going to be passed through the pores, and the larger size cells, they are going to be retained over the filter paper, membrane filter paper. How it is chosen? How the selection it is there? What has to be retained over the filter paper? The mature cell. The cell which has the larger size, it has to be retained here. It has to be retained over the filter paper. And the cells which are of the smaller size than the mature cells, they are going to be passed through the membrane filter. Any sense? Okay. Okay. So they are going to be passed through what? Filter paper. So here there is the addition of the culture. Allow it to filter through the membrane filter. Okay, so the smaller cells, now they are going to be in this medium. Okay, the smaller cells, they are going to be in the medium, and who will be retained over the membrane filter? Who? Mature cells. Okay, now here in the second step, what is going to be done? The membrane filter, now it is going to be inverted. Okay, the membrane filter, it is now inverted. Okay, so the mature cells, they are on the downward size, and now here you are going to add who? The medium. As the medium you are going to add here on the mature cells, as that they are the mature cells, what will happen? These cells they are prone to divide. So what will happen to these mature cells? They are now going to divide. Okay, mature cells, it is going to divide into the two daughter cells. Now what you have added? You have added the membrane, you have added the medium. So as you are going to add the medium, these mature cells, now they are going to divide, and it is going to be form what? It is going to form the daughter cells. Now here the size of the daughter cells, it is what? It is It is a smaller than who? The mature cells. And now here, as the mature cell, it is going to divide. All the mature cells, they are going to be divided. The population of the cells you are getting here, that population of the cells, it is of the, it is a homogeneous. That is, all the cells, they are in the same life cycle. They are all the daughter cells. They are of the same size, same age. And this it is going to be called as what? This it is going to be called as a synchronous culture. Drawback of this method, drawback of this Hemsteller and Cummings method or the technique, it is what? The smaller size of the cells, that is the small number of cells, they are going to be collected. So what is the next then? What is the next method? Okay, the next method, it is the density gradient centrifugation. Now when in the medium there are the mature cells, there are the daughter cells, or there are some of the cells, they are about to mature only. Okay, they are not dividing, they are just getting mature one. So the medium, it is how? The heterogeneous medium. Now here, heterogeneous culture, it is there. So the cell, it is of the larger size. Its cellular constituent, it is a larger one. So its density, it is going to be larger one. These are the daughter cells. Cellular constituents, it is a smaller one. So its density, it is going to be a smaller one. The intermediate, it has a cellular constituents in between the mature and in between the daughter cells. So what is happening? Its density, it is going to be a different. So here, according to the size and the age, the density of the cells, density of the bacterial cells, it is going to differ. And this deferred density, okay, this deferred density, it can be, okay, deferred density of the cells, it is used, okay, it is used for the separation of the cells according to their size and age. Now here, when in the density gradient centrifugation, the gradient, it is made in a such a way that the cells, they are going to separate according to what? According to its density. Okay, according to its density. So here you are going to maintain the density. You are going to maintain the density gradient. Your medium, the culture, it is going to be added. It is now going to be in a density gradient centrifugation. And what you observe? You are observing the number of bands here, and this number of bands, it is nothing but it is what? It is the bands which has been obtained by the bacterial cells. Now here, this it is having different density, this it is having different density. That is, the cells, they are separated according to what? According to their densities. Largest density, mature cells, then the intermediate cells, then these are what? These are the daughter cells. So in this way, there is what? There is a separation of the bacterial cells according to its density, and its separation according to the density, it is ultimately the separation according to the size and age, because at every size, at every age of the cells, the cellular constituents, it is going to differ. Okay, the mass, it is going to be differ. So its density, it is going to differ. That's why here in the density gradient centrifugation, it is going to be what? It is going to be separated. Okay, got this? Now the next one, selection by induction. What is there? It is the selection by induction. Here there is a use of the shock treatment. Shock, it has been given. Shock of what? Shock of temperature. Or there is a shock of, or here there is a use of what? There is a use of starvation, sublethal doses of the radiation or the drugs, or there may be the use of the exposure to the light. When the bacteria, it is a photosynthetic bacteria, at that time there is a use of what? Exposure to the light. Here we will look at the temperature. Okay, induction, selection by induction by using the temperature. Okay, that is the shock, it has been given, it is of the temperature. Now suppose here we will take the bacteria, it is E. coli. Now E. coli, it has an optimum temperature at 37 degrees Celsius. Optimum temperature means what? A temperature at which, optimum temperature means what? A temperature at which, okay, the bacterial cells, they are going to grow fast, or the maximum growth, it is going to be observed. Okay, the maximum growth, it is going to be observed at a temperature. That temperature, it is called as the optimum temperature. Okay, so the 37 degrees Celsius temperature, it is optimum for the E. coli. Means what? The highest growth rate, it is going to be observed, the maximum growth, it is going to be observed at a 37 degree Celsius. Okay, Now as you are going to decrease the temperature from the optimum, the growth rate, okay, the growth rate, it is going to what? It is going to decrease. Okay, so there will be such a temperature at which there is a no growth. Now in the microbiology, growth, it is called, growth means what? Increasing cell number. That is a cell, it is going to divide from the one cell to the two cells, that it is called as an growth. So at the temperature, it is going to be lower down, the growth, it is going to be decreased, that is the number of cells, they are going to decrease, and at a particular temperature you are going to observe that there is a no growth. And that temperature here, some of the time, it is what? It is a 4 degrees Celsius. 4 degrees Celsius here, at the 4 degrees Celsius, 4 degrees Celsius, most of the time at the 4 degrees Celsius, we are going to preserve this E. coli. Okay, at a lower temperatures, okay, at a lower temperatures like the 4 degrees Celsius temperature, the E. coli cells, okay, here the E. coli cells, they are not able to grow. What is happening? The E. coli cells, they are not able to grow. Then what is happening to the E. coli cells? They are going to increase the cellular constituents. That is, they are growing only, but here they are going to increase the cellular constituents. Suppose this, it is an daughter cells, okay, that's our daughter cells. Now it will become a mature cell by increasing the cellular constituents. That is, the at 4 degrees Celsius, growth, it is not going to be there, cell division, it is not going to be there, but what is going to be there? There will be the metabolic activity, it is going at a lower level, and that metabolism, it is allowing a daughter cell to become a mature cell, but not allowing this mature cell to get divided, because the temperature, it is not a temperature for the growth, that is for the division. The temperature, it is not a optimum temperature. Got this? Now this, it is used, okay, this logic, it is used to obtain what? To obtain this synchronous culture. Now here the E. coli culture, it has been taken, okay. Now what has been done, for half an hour, okay, for half an hour the culture, it is going to maintain at 37, in the next half 4 degrees Celsius, in the next half 37, next half 4 degrees Celsius. Alternatingly, there is what? There is a shifting of the temperature. So here suppose the first, it is a 37 degrees Celsius for the half hour. There is keeping the culture at a 37 degrees Celsius. Then what you observe? There is a growth. That is, there is a logarithmic growth. And now you are going to shift the temperature from 37 to the 4 degrees Celsius. You observe that there is a no growth. The cell density, cell number, it is not going to increase. Why? Just I have explained, at 4 degrees Celsius, metabolic rate, it is lower down. The 4 degrees Celsius, it is not a temperature at which there is a cell division. So the cells, okay, the cells, they are just going to mature. They are not going to divide. They are not going to divide. And as they are not going to divide, here what you observe, that there is a no increase in the cell number. Okay, then there is a shifting of the temperature from the 4 degrees Celsius to the 37 degrees Celsius. Then what will happen? Tell me what will happen? The cells, now they will grow. All the cells, okay, all the cells which are present at 4 degrees Celsius, how they are? They are mature one. At 37 degrees Celsius, they are getting the optimum temperature. So what will happen? The cells, they are going to divide. And as the cells, they are going to divide, what you observe? The cell number, it has been increased. Are you getting? The cell number, you are here, it is the cell number, it has been increased. So you are going to observe that there is what? There is a log phase. Again, you are going to keep at where? 4 degrees Celsius. Again, there will be the lag phase. Why lag phase? Because these daughter cells, okay, these daughter cells, they are requiring the time to get mature, and at 4 degrees Celsius, it is not a temperature for the cell division. So just the daughter cells, they are going to mature. Again, at 37 degrees Celsius, what will happen? You are going to observe that there is what? There is a log phase, that is an exponential phase. So in this way, in this graph, what you observe that there are only the two phases alternating to each other, and that phases they are lag phase and a log phase. Which two phases? Lag phase and the log phase. Lag phase, exponential phase. Lag phase, exponential phase. Lag and the log phase, they are going to alternate to each other, and such a type of an graph which has been obtained, that it is called as an synchronous growth curve. What it is called? Synchronous growth curve. In that, you are only going to obtain the two phases alternating to each other, lag phase and a log phase. Okay, and such a type of an phases when you are obtaining, okay, at that time you are going to say that you have obtained what? You have obtained the culture, and that culture it is going to be called as an what? Synchronous culture. What it is called? It is called as a synchronous culture. Same, it can be done for whom? Starvation, sublethal dose of the radiation drug and exposure of the light. In the starvation, there is starvation of the essential nutrient. As essential nutrient it is not there, what will happen? The cells, they are only going to mature. As you are going to provide the complete medium, cells, they are going to divide. Okay, next again you are going to starve the cells for the essential nutrients, so again the cells, they are going to mature. In this way, there is what? Again you are going to observe that there is a lag phase and a log phase, lag phase, log phase, and in this way, by observing that is here, as you are getting the synchronous growth curve, you are going to say that your culture, it is what? The culture, it is what? Culture, it is a synchronous culture. Okay, have understood what do you mean by the synchronous culture? Synchronous cultures are the cultures in which all cells, okay, all the bacterial cells, they are in their same phase of life cycle, that it is called as a synchronous culture. And the graph which has been obtained during the obtaining the synchronous culture here, in which there are only the two phases of their life cycle, lag phase, log phase, lag phase, log phase, that is the alternating lag and log phase. Such type of an growth curve, it is called as a synchronous growth curve. I think you may have cleared the idea about the synchronous culture. If you like my video, please subscribe my channel and also share my video with your friends, so the knowledge it is going to spread. Thank you.